This lecture is all about one category of macromolecule, specifically carbohydrates. And carbohydrates are based on sugars, and they are going to be important calorie sources for animals as well as including humans. And so they are going to give us things like bread and pasta and potatoes and corn, for example, or at least they constitute a large portion of those various food items. And this picture is making me hungry already. In this lecture, we'll start by going over the general formula for carbohydrates. We will then make sure you know the difference between three categories: monosaccharides, disaccharides, and polysaccharides. And we will go over some more specific naming for monosaccharides that will tell us how many carbons are in each. We will talk about how polysaccharides and disaccharides are formed by the process of dehydration synthesis, and we will end by going over some major categories of polysaccharides. As we already mentioned, carbohydrates are found in items like grains, fruits, and vegetables. We'll talk about some different roles they serve in these. Glucose is one kind of carbohydrate. There are also other carbohydrates that are not based on glucose, but glucose will be very important when we talk about respiration. The general formula for carbohydrates is shown here, and this is something you need to memorize. What it shows is, in parentheses, there is C, H2, and O. This tells us that the ratio of carbon to hydrogen to oxygen is always going to be 1 to 2 to 1, twice as much hydrogen as carbon and oxygen. However, the number of each of these can vary according to N. So N could be a number from 1 through 6 for the ones that we will be thinking about. And so if the number was 3, for example, that would mean the formula was C3H6O3. If we were thinking about a ribose, which has 5 carbons, the formula would be C5H10O5. And if we think about glucose, then the formula would be C6H12O6. So the numbers can vary as long as the ratio is the same. We'll see that this formula is the ratio for monosaccharides for single subunits. When we start putting together disaccharides and polysaccharides, which have multiple subunits, then the ratio is going to change a little bit because of dehydration synthesis, and we'll talk about those details when we get there. There are three main categories of carbohydrates: monosaccharides, disaccharides, and polysaccharides. The difference is that monosaccharides have only one subunit, and so that's what the prefix mono is telling us here, mono meaning one. Disaccharides then have two subunits, with the prefix di meaning two, and polysaccharides have 3 or more subunits, with the prefix poly meaning many. In each of these cases, saccharides is simply referring to the fact that these are sugars. Let's go on and talk a little bit about monosaccharides. Monosaccharides are the monomers of carbohydrates, so they're the repeating subunits. They usually have 3 to 7 carbons. The ones that we'll talk about always have between 3 and 6. We will see that monosaccharides and disaccharides end with the suffix ose, so OSE. And we'll look at some names of those in just a minute. A common feature of monosaccharides is that they include a carbonyl group, at least when the monosaccharides are in their linear forms, as shown in these diagrams. So a carbonyl group is a carbon double bonded to an oxygen. And you can see in the diagram at the bottom right 3 examples of where that carbonyl group is in 3 different monosaccharides that we will talk about during the semester. So our monosaccharides will end with -ose. We can have some general names for categories of monosaccharides that tell us how many carbons there are in it. And so a 3-carbon monosaccharide is generally called a triose. Tri meaning 3 and telling us there are 3 carbons, ending with ose telling us it's a sugar. 5-carbon monosaccharides are generally called pentoses, the prefix pent meaning 5. And 6-carbon monosaccharides are generally called hexoses, the prefix hex meaning 6. Now, each of these are general categories, and within them there are a variety of different monosaccharides, depending on how the carbons, oxygens, and hydrogens are arranged. So we'll see that there are multiple kinds of pentoses and multiple kinds of hexoses, for example. So here's a more specific example of hexoses. Remember, hexoses have 6 carbons, which you can count in this diagram. But there can be isomers of hexoses. So any hexose would have to follow the formula C6H12O6. And if you go back to the general formula for carbohydrates, you can see why that is. But you can see in this diagram that the location of the carbonyl group, for example, is different between the first 2 and the second. And between the glucose and galactose, the first 2 diagrams shown here, you can see some other differences in the arrangement of hydrogens and hydroxyl groups. So since all 3 of these have the same formula, C6H12O6, but they have different arrangements of where the atoms are in the molecules, these are called isomers. And we talked about the general term isomer back in chapter 2. You do not need to memorize the formula, or you do not need to memorize the specific shapes of glucose, galactose, and fructose. You should just recognize them as being isomers. The one of these that we will talk about most is going to be glucose. We will move on now and talk about how monosaccharides can be combined to form disaccharides, or dimers. And this process, just like for other macromolecules, is going to happen through dehydration synthesis. And so remember from our general introduction that dehydration synthesis links monomers onto other monomers or onto chains, and it does so by releasing a water molecule. So in this diagram, we happen to be showing the linking of glucose on the left and fructose on the right. These are both hexoses. They're both 6-carbon sugars. And when they combine together, they are going to form a disaccharide called sucrose. And you're all familiar with sucrose as table sugar. And so this is what you would use, for example, to sweeten coffee or if you were baking. You can see on the diagram the hydroxyl group on the glucose and the hydrogen on the fructose that are going to be combined together to form a water molecule when dehydration synthesis occurs. And you can also see that this oxygen, which used to have 2 bonds, one up to this carbon and one over to this hydrogen, it is now going to be, it will only have 1 bond, and it will need to form a second covalent bond. It will do so by linking all the way over here to this carbon that used to have 4 bonds, one here, one here, one down to the hydroxyl, and one to this carbon up at the front. But when the hydroxyl leaves, this carbon will have only 3 covalent bonds. It will need to form a 4th. So by this oxygen linking to this carbon, both the oxygen and the carbon can fulfill the octet rule. So let's watch this process occur. When the water molecule leaves, we now have this bond formed through the oxygen. The oxygen has the 2 covalent bonds it needs. The general name for this bond that is linking monomers of sugar molecules together, or generally linking monosaccharides together, is a glycosidic bond, which is the same thing as a glycosidic linkage. We'll use the word linkage and bond interchangeably. So this bond has formed, and now our two monosaccharides are a disaccharide, and this one happens to be specifically sucrose. Sucrose is only one of many possible disaccharides. Some other examples of disaccharides are maltose, which is used for making beer, for example, lactose, which is a common sugar in milk, and sucrose, which we already talked about. So you don't need to memorize these different structures. I just want you to see here that even though each of these is formed by two hexoses combining together to make a disaccharide, these structures are different between all three of these molecules. In all cases, the bond that is linking the monosaccharides together is still a glycosidic linkage. We've talked about linking two monosaccharides into a disaccharide. As we start adding more monosaccharides onto that disaccharide, then we will have a chain of 3 or 4 or possibly many monosaccharides joined together. And we will use the term polysaccharide for these. So while technically this only has to be at least 3 monosaccharides long, typically this is a very long chain of monosaccharides. And as we've already said, they will be joined together by glycosidic linkages. These chains might be branched or unbranched, and they might have only one kind of monosaccharide, or they might have multiple kinds of monosaccharides included. And there's an example here. You can't see individual monosaccharides in this. This is showing the inside of a cell, and these darkly stained areas are glycogen, which we'll talk about in a bit as being one important kind of polysaccharide that is used for energy storage in animals. The final part of this lecture is going to be reviewing some important kinds of polysaccharides. And we are going to start with starch. Starch is used by plants for energy storage. So I wrote here in plants, but it's important that this is for energy storage because we'll contrast that with a different polysaccharide in plants momentarily. In starch, it's always glucose molecules that are being joined together, and they're being joined together in a particular configuration that links the number 1 carbon in one glucose with the number 4 carbon in the next glucose. And we'll go over the numbering of carbons in just a minute. Since it is linking the number 1 and number 4 carbons, we are going to call this bond a 1,4 glycosidic bond. And this particular configuration is called alpha. And so specifically in starch, we have an alpha 1,4 glycosidic bond. So let's talk about how carbons are numbered in glucose specifically. In glucose, the way to figure out which carbon you're looking at is to find the oxygen in the ring structure here, and then proceed clockwise from the oxygen. So anywhere there's an intersection on a diagram like this, it is implied that there's a carbon there. But because organic chemists work a lot with carbon, and because every carbon forms 4 bonds, it gets really hard to write C's in at these intersections. And it's easier to represent it simply by having an intersection with no letter there. So if it's easier for you, you can go back and write a C in at any of these unlabeled intersections. So there's one here, a carbon here, a carbon here, a carbon here. And then this last carbon, the illustrator chose to write in just to make it a little bit easier to look at. But they could have equally left this out if they had wanted to. So proceeding clockwise from this oxygen, we have carbon 1, 2, 3, 4, 5, and 6. If we go to our next molecule, the numbering is the same, which is how we come up with the numbers 1 and 4 for these carbons. Notice here that all three of these glucose molecules shown are in the same orientation. So in each of these cases, this number 6 carbon that is not part of the ring structure is sticking out towards the top of the molecule, as depicted in these diagrams. This particular relationship happens to create a polysaccharide that is going to form a coiled or helical structure. So you could picture along this hundreds or thousands of glucose molecules, and they would slowly coil around. That means that starch inside the cell is a tightly coiled structure, and that's going to be important for the cell because it limits the amount of water that needs to interact with all of those glucoses that are being stored. Glucose is a hydrophilic molecule, and by condensing all of these glucoses in a chain without much water interactions, then the glucose in the cell is not going to disturb the water balance within the cell. And so if we were to look inside a potato, for example, then we would see a lot of starch in this coiled form. A second very closely related molecule occurs in animals. So this is glycogen, and glycogen, just like starch, is linked it links glucoses together, and they are linked together by alpha-1,4 glycosidic bonds. So that's exactly what we saw for starch. These are very similar molecules. The difference with glycogen is that glycogen has some glucoses for which a branch occurs, and instead of having only one glucose attached, it has two glucoses attached. And so you can see that these occasional branches in glycogen give it a different shape than we saw for starch on the previous diagram. Glycogen is used for energy storage in animals, just as starch is used for energy storage in plants. Next, we will talk about cellulose. And cellulose is shown here. Unfortunately, I made a misspelling, so there should be an L right here between the U and the O. So please add that. Cellulose is again a polysaccharide. It is again a polysaccharide of glucose. And in this case, it is used for structure rather than energy storage. So it helps plants stand up. It helps cell walls avoid rupturing. Let's start by looking at the molecular structure of this, and then we'll look at its shape within the cell. On the molecular level, this is still being made from glucose, just like we saw from starch. And for the glucoses for which that number 6 carbon is pointing upward above the ring, we can still number them clockwise from the oxygen. So here's our oxygen. We would still count them 1, 2, 3, 4, 5, and 6, just as in a glucose and starch. However, the new twist here is that every glucose monomer, every other one is flipped in orientation. So the second and fourth have this number 6 carbon pointing upward above the ring, while the number 1 and 3 have the number 6 carbon pointing downward below the ring. The structure of the monomer is the same, it's just that the orientation has been flipped 180 degrees. So now if we want to count the carbons, because it's flipped over and we're looking at its bottom, we have to go in the opposite direction. So we would count these as 1, 2, 3, 4, 5, and number 6 coming out of the ring. The linkages in cellulose are still alpha-1,4 linkages, because it is still a number 1 carbon and a number 4 carbon that are being attached through this glycosidic bond or glycosidic linkage. However, because it's a different orientation of the glucose monomers, we are going to call this the beta-1,4 glycosidic linkage instead of the alpha-1,4 linkage, as we saw in starch. So in starch, it was alpha-1,4, In cellulose, it is now beta-1,4. And even though this seems like a very small change, what this does is it changes the structure of this molecule in the cell. Instead of it being a coiled molecule or a helical shape, as we saw for starch, it is now a linear shape. This is important because if the plant puts this in the cell walls, then this linear shape gives a lot of rigidity and a lot more strength than a coiled shape would give. A coiled shape like a spring could stretch, but a linear shape like this will not stretch, and that will allow this to be a more useful structural material for the plant. We now are going to talk about 1/4 category of polysaccharide, and this is chitin. And chitin is actually a modified polysaccharide because it doesn't strictly follow that formula we've seen of CH2O and then repeating units of that. Instead, chitin also contains nitrogen. And the way I remember this is simply that chitin ends with an N, and nitrogen, of course, is symbolized by the letter N. And chitin, like cellulose, is used for structural support rather than energy storage. Specifically, it makes up the hard exoskeleton in arthropods, things like, crabs and spiders, and insects, such as the bee shown here. It also creates the rigid structure in mushrooms and other fungal bodies. So you don't need to memorize the specifics of this structure, but you should see that generally this looks like a carbohydrate with carbon, oxygen, and hydrogen in approximately, although not exactly, the 1:2:1 ratio that we'd expect. But you can see there's a nitrogen here and a nitrogen here, so it is a more complex molecule. Let's end by reviewing general important lessons about carbohydrates. First, the monosaccharides follow a general formula of having carbon, hydrogen, and oxygen in a 1:2:1 ratio. Second, the monomers or monosaccharides can be categorized by the numbers of carbons they have, with Latin prefixes like tri, pent, and hex telling us how many carbons. So a hexose, for example, would have 6 carbons, 12 hydrogens, and 6 oxygens. Within each of those categories, there can be different arrangements of the atoms, and so there can be isomers. Number 3, we can link monosaccharides together into either disaccharides or polysaccharides through the process of dehydration synthesis. We could also remove monosaccharides through the process of hydrolysis if we wanted to shrink a polysaccharide or split a disaccharide. Fourth, glucose is a very important monosaccharide. It is a 6-carbon sugar, or a hexose, and it can be connected together to form starch, glycogen, or cellulose, or it can be modified in order to form chitin in arthropods and fungi.